So I'm Eija Korpelainen from CSC, and I'm going to tell you about two approaches that we have used in single-cell RNA-seq training. So first, a little bit about our case. CSC is a national supercomputing center, and we are also like the National Bioinformatics Core Facility. So we have a lot of users from all the universities around the country. Now, a large number of our bioinformatics users are non-programming scientists with either medical or biology background. On the other hand, we have very limited trainer resources in bioinformatics, and now we are facing this challenge that we need to teach the very fast-moving and rather complicated field of single-cell omics data analysis. So the problem is twofold. So on one hand, the users are not able to use R or Python, and on the other hand, the analysis theory is quite advanced. So we found that when we were trying to cram this kind of things to people's heads in just a couple of days, the end result was more like this. And we obviously cannot go this way, so we had to come up with some other approaches. And this is what we find that helps us. So first of all, we provide user-friendly web-based access to the analysis tools, and I will be talking about that shortly. We use the flipped classroom approach in our courses, and we have also benefited greatly from both national and international collaboration. So first of all, about this flipped classroom. So what that means is that essentially people listen to the lectures at home, and then when they come to the course, then we do exercises and discuss, and they can ask questions. So the good thing about this, especially in single-cell RNA-seq training, is that uh, it allows people to swallow the theory in small doses, so they have time to digest it. So what we do is we make short lecture videos on each topic, and then we give those videos to the course participants already 2 weeks before the course, so that they have time to view them at their own pace. And so this goes both for the virtual courses and face-to-face courses. Now then, during the actual course days, we recap the theory on say topic X, then we do exercises on that topic, check the results together and discuss, and then we move to the next topic. So the theory and practice is interleaved. We also provide a Google Doc where people can write questions and comments, and of course, they also ask directly with their mics during the course, but this way the questions and answers remain so that it's easy to go back to them. And what I should also say is that when we give the lecture videos, we actually also give them already then a set of theory questions so that they can check if they have understood things or if they need to watch the lectures a bit more. So as an end result of this, we find that people come to the courses well prepared and they ask very valid questions. And the course time can be used for discussions and exercises and questions, so we can use it more efficiently. Uh, an additional good thing about it is that we can also provide links to more advanced lectures by others in addition to the ones that we made. And there I would like to thank very much Paolo and Ahmed, since we have actually their lectures from our previous courses. So then moving on to the web-based platform that we used. So this is the Chipster platform that we have developed. And the good thing there is that people can operate it with mouse. So because we avoid the technical hurdles, then we can really focus on the theory during the course. I don't know if it's appropriate for me to say this, but in some ways, this is like the drug dealer approach. So we get people addicted using this easy way of doing bioinformatics. They like it, and then we see some of them moving to harder substances like R and Python. We do provide training accounts to Chipster, so people from other countries can use it as well without setting up the server themselves. What is also handy for using this during the course is that Chipster users can easily share analysis sessions within the software, and the software also tracks the metadata, so like what tool was used with what parameters and so forth. So this helps us to troubleshoot easily during the virtual courses when we can just look at people's sessions. And what I should also say is that, of course, this system also allows people to see the source code, like so the R code, for example, if they wanted to. Here are some technical details. So we provide this installation package, and we also help institutes to set up their own servers. So this is what it looks like. So here I have been doing some analysis. So this workflow view shows the different steps that I have done. Here are the analysis tools, and people can also visualize their results. So it seems to work quite well. This is just one comment from one user who was actually able to use R, but felt that he got actually further using this system. Then finally, I would like to highlight the importance of international collaboration. So here I have two examples. I hosted a course in 2019 where we had trainers from 6 different countries. The lecture videos were recorded, and so far the playlist has over 33,000 views. So, really, when you give lectures, please record them because this way um, many more people can actually benefit from them. Otherwise, there's another example. So, the Elixir community for single cell omics that we are now in the process of setting up. There are roughly 20 countries involved. We are in the process of preparing the roadmap, which hopefully will be published as a white paper in 1000. And there will be heavy emphasis on collaboration, on training, in addition to other things like tool benchmarking, etc.